Morning, everyone. Once again, I am with a new video, and this time I am discussing the next topic, which is nucleic acid A1.2. And we are already, I have started a series of video on the latest syllabus of IB biology. The first assessment will be in 2025. So again, I want to welcome everyone, and uh, in today's session we will talk about the content of the topic, and we'll go into the detail of the subtopics also, and we'll also cover SL and HL section of that particular topic, and at the end of the session I've also planned to upload some question banks related to paper 1 and paper 2 type, so that you can go through it and you can use it for your lessons, and student can work on the questions to get good grades in coming exams. So without delaying, let's start with the topic. So the plan is to start A1.2 nucleic acid, which is a very important question, the topic, as in terms of the subject, and in terms of your board examination also, like major questions, connecting questions you will get from this particular unit. So this is the syllabus guide I have taken from the subject guide, and which shows standard and higher level have 3 hours of lesson, and additional higher level needs 2 hours of lesson. There's a guiding question. So let's go into the detail of the topic. A 1.2.1 talks about DNA acting as a genetic material. So in this case, we will talk about the basic idea about DNA and RNA as a genetic material. Then we will go into the detail of the structure. We'll talk about the nucleotide of this structure. Then we will talk about the backbone, how sugar phosphate bonding takes place. This is your sugar molecule, this one, pentose sugar. This is your nitrogenous base, and this is phosphate. So how they are connected, we'll talk about that. We will also talk about the base present in the nucleic acid, which forms you can say a kind of information which makes your body. We'll also talk about how the information are coded. Then next we will talk about how RNA act as a genetic material. What is the basic component of the RNA? How they are made up of, with a basic diagrammatic representation. After that, we will talk about why DNA is double helix, how we can consider them as an anti-parallel strand, and how the bonding takes place between both the strand. As you can see in this particular diagram, the cytosine connects to the guanine and the thymine connects to adenine. We'll talk about these in this section. Then we have difference between the DNA and RNA, right? We'll talk about in detail again. What are the differences? Role of complementary base pairing in the process. Then we'll talk about diversity of the possible DNA-based sequences, which gives a limitless capacity of DNA to store the information. So we'll talk about how and what characteristic feature of the DNA makes them like a big cloud of the information, the data which is being stored inside it. And we'll talk about the diversity connecting to the DNA lengths and the base sequence possible, right? And we will also talk about how the DNA is acting as a storage for the information which makes our body. Then we will talk about conservation of the genetic code across all the life forms, which maintains the evidence of universal common ancestry, like we have studied in so many books regarding we all come from a common ancestor. So we'll talk about that also, how DNA is giving an evidence that yes, we are emerging from a common ancestor. Then we will talk about the additional HL part of the topic, which consists of directionality of the DNA and RNA molecule. We'll talk about the linkage, 5-3' linkage in the DNA and RNA molecule in terms of DNA replication, in terms of DNA transcription, and in terms of translation. So where the linkage is going on, we'll talk about all these things in detail, but we are not going into the detail of the process. Rather than we will see how 5 to 3 linkage is going in the sugar phosphate bone in terms of in terms of replication process, transcription process, and translation process. Next topic consists of purine to pyrimidine bonding as a component of DNA helix stability. So why for stability, for DNA structural stability, we need AT bond, CG bond, the base pairing. Why it is like that? What makes the structure unique after this, right? We'll talk about that in the 3D model. Next topic talks about how the DNA is confined to a very small region in the nucleus, and we'll talk about the nucleosome model. After that, we will talk about the evidence which proven that DNA act as a genetic material. It was an experiment done by Huxley and Chase, right? So we'll talk about that experiment, and then we will talk about the Chargaff data on the relative amount of pyrimide and pyrimine bases across diverse life form. So there are some nature of science topics. We'll discuss that also with the help of the experiment done by these scientists. So, and most important, the linking question, which is very important. They have incorporated this new concept in the syllabus, how we can connect our current topic with the another topic, right? So here they are talking about what makes RNA more likely to have been the first genetic material rather than DNA. Second is how can polymerization results in emergent property. We'll talk about that also in the lesson. So let's begin with the content. Welcome everyone once again. I am your host. I'm going to discuss about all this topic in this video. Stay connected. Let's begin. So the first topic talks about the DNA as a genetic material for all the living organisms. How and why? Let's see. So you will find that inside the cell, there are some biological molecules which is always present, right? When we say biological molecules or biomolecules, we are focusing on the things, the living molecules which are present inside the living system, right? Like carbohydrate, lipids, proteins, and nucleic acid. In this particular section, we will discuss about the nucleic acid. So you can see carbohydrate is given in B1.1.4, lipids are given in another topic. So wherever you are finding these quotations, this number, this shows the topic number, topic name and the number, right? So it will be easy for you to navigate. So the right side of the diagram shows the carbohydrate. You can see this is hexagon hexose sugar. Both are joined to make a disaccharide called lactose. This is protein molecules. This is your nucleic acid. There are so many things attached. Let me tell you one by one with all the diagram. This is your adenine combined with the pentose sugar, and this is phosphate. So this is adenosine monophosphate, right? Like this is GMP, TMP, CMP, and UMP. So these are the nucleotides. And at the end you can see the lipid molecules, and they have a long carboxylic acid chain, hydrocarbon chain, and you have a carboxylic acid end, right? So these are the basic biological molecules present in the DNA, present in the nucleus of the cell, right? And not only the nucleus, it is present everywhere in the cell, right? And they all are found in the living body. When we talk about how the nucleic acids are formed, so you will find that there is a series of reaction. We'll talk about the reaction later on. Before, we'll try to understand the function of the nucleic acid, what they are doing, what is the purpose. So majorly they have two primary functions. The first function is to pass the information from one generation to the next generation, so that the flow of information should be unique. Information at any point of time should not change, because if there is changing, it will may cause mutation or any genetic abnormality in the next generation. So it's become very integral process for a cell to make exact same copy of DNA without disturbing the any base sequences in the DNA. So that is done by the DNA replication process, which is very important, in which one DNA will make another DNA copy, and the mode of DNA replication is always semi-conservative. The meaning of semi-conservative is, you can see this particular DNA is the old DNA and the strand is open, right? And the new strand is formed, and you can see the base sequence are exactly matching with both. You can see GC, GC, TA, TA. So base sequence is not changed. And in all the new DNA which is formed, you will get one old DNA and one new DNA. So we can say that old DNA is maintaining a template for the another strand to bind. So if there will be A, of course it will bind with T. So in this way they are maintaining consistency in the base sequence, right? So through this process the information gets stored as well as it is transmitted to the next generation, which is very important. And at the same time, the next main function of the DNA is to produce protein. So DNA is making RNA copy, RNA makes protein, that we all know. But overall, why the DNA is stored? The answer is to make protein. So the information about what type of proteins are going to be formed by the cell is controlled by the DNA. So the sequence of nitrogenous base which you will see in the DNA strand, they have the information that they have information in a form of a code, genetic code, that helps to make protein in the process. And the process is called translation. And after translation, these proteins are customized, modified, and regulated in such a way that they can be expressed in the body. That's called gene expression. And there is a limitless capacity of DNA to store information. It's limitless. You can't concise or constrict the information. It's very vast. So these are the two main functions. When we talk about the type of nucleic acid, so majorly we have two types. First is DNA, which stand for deoxyribonucleic acid, and whereas the RNA molecule is a single strand molecule, which is stand for ribonucleic acid. Both have the same function. First function, they act as a genetic material. They pass hereditary information between the cells, between the generations of the cells. And the second most important function was to make a protein. So they have the coded information which helps to make RNA also. So DNA makes RNA, that is possible. And the process by which this DNA molecules makes RNA, we call them transcription. Whereas the RNA ribonucleic acid, first function, as I said, it's act as a genetic material. Second function, there are some non-genetic RNA, such as you can see here mRNA, rRNA, and tRNA, stands for messenger RNA, ribosomal RNA, and transfer RNA, which helps in protein synthesis. So both DNA and RNA have different functions in our body, but they may act as both are acting as a genetic material at the same time, but in a cell, any one will act as a genetic material. And the DNA which is making an RNA molecule inside the body, inside the cell, They are not genetic RNA, it's a non-genetic RNA. Example, you always remember messenger RNA, ribosomal RNA, and transfer RNA. Well, this is the basic idea about the two types of the nucleic acid we have in our body. This is the group of scientists. They are the two people who have proven that DNA is a genetic material. Is Martha Chase, born in 1930, died in 2003. Alfred, born in 1908, died in 1997. Both have a credit. They have done an intensive experiment to demonstrate that DNA acts as a genetic material. So we'll discuss about the genetic, how and what kind of experiment they have done to prove that DNA is acting as a genetic material. It will come later on in the slides. Let's go to the next section. So this was basic idea what they have done. They have taken a bacteria and they have taken a virus. When virus infect bacteria, they mix bacteriophages. So these are the bacteriophages. So in the bacteriophages, they have taken protein, they have taken the DNA content of the bacteriophages, and both are radioactive labeled, and then later on they have checked which is coming to the next generation. So of course we know now that protein is not a genetic material, so protein is not going to the next generation. But if you have radioactively labeled the DNA and DNA is acting as a genetic material, so of course they will go to the next generation, right? Because in the culture you will grow the bacteria, and once bacteria will grow, you will find the same genetic setup is going to the next generation. So if the DNA content of the previous generation was radioactive labeled, you will find that the same radioactive labeled DNA content is going to the next generation. If you have a question why, because of the semi-conservative mode of DNA replication, means the new DNA which is formed, they have half old content, half new content. So in this way, we'll see separately how he has done the experiment. This is a basic representation what he has done. So we'll move to the next slide and we'll come back to this again, where we will discuss more in detail about the process. Next, DNA is a universal to life. Now when we say universal, means it's very characteristic feature of any living organism, right? So although RNA is hypothesized to be the first genetic material evolved, that was an hypothesis, but today all living organisms use DNA as a genetic material. But of course we can't deny there are some other viruses, some like reovirus, retrovirus, poliovirus, AIDS virus, HIV virus, coronavirus, they all have a RNA as a genetic material, right? So there are some viruses in which RNA act as a genetic material. So the use of the genetic cross code across all the form of life is actually gives an evidence of universal common ancestry. And the sequence of DNA in cells can be used, analyzed, compared to find the relationship between any species or any organism. So most similar the sequence means they are more closely related. That's how they can connect to the ancestor, and they can find out like how much similarity. More similarity means more closely related. So the biological similarity of all the current life suggests that the last universal common ancestor of all the life used DNA as a genetic material. So that is now proven. So DNA is the very oldest genetic material on Earth. Yes, we can't deny, as I have given some example, we have some viruses, they have DNA, some have other nucleic acid as a genetic material like RNA molecule. So some viruses use RNA as a genetic material. However, some viruses are not made of cells, so we can't consider them into this living category, right? Because they are not cell, right? It's just a DNA molecule which is covered by a capsule kind of covering, which is made up of protein, and that where so there is no cellular characteristics or the cellular property where we can suggest that this is a cell. So if virus is simply present, they are not living because they are not inside the cell. Once they go inside the cell, they use the cellular machinery and they behave like a cell, right? But normally they are not a cell. They are exception to the cells. Let's talk about the component of the nucleotide. Nucleotide is a monomer unit. Monomer unit means the single unit, one unit, like one pen. You have so many pens, right? So many pens is polymer. Single pen, monomer. In the same way, nucleotide is a monomer, and when we add all the nucleotide in a long chain, we call them polynucleotide. So it's now a polymer unit. It's a big unit. So in this section, we'll talk about the single unit of the DNA, and we'll see how it is made up of, what are the components present in the nucleotide. Well, you can see there are three basic structures. First is the base, which is also called nitrogenous base. This is pentagon structure to symbolize pentose sugar, right, which is deoxyribose sugar in case of DNA. And this circular part is phosphate, and you can see that the base is connected to this particular end. This is very important. Whenever you are drawing a nucleotide, a single molecule, you must connect the base to this particular corner. So we can consider this as the first carbon. So if I am saying the number, it will be first, this will be 2, second carbon, this is the third carbon, fourth carbon, and the fifth carbon, which is connected to the phosphate, right? So this you have to take care while connecting the base square to the pentagon and pentagon to phosphate. So the bond which is present here, this is called covalent bond, also called glycosidic bond, whereas this particular bond is also called covalent bond, and you can also call it as a phosphatized bond, right? So names you have to remember, right? So this particular bond, this bond where my cursor is, this is called covalent bond or glycosidic bond, glycosidic. This one, which connects phosphate to the pentose sugar, this is called phosphatized bond. You must remember this. Here I have given you the number, so you can see how the base is connected to the first carbon of pentose sugar always. Example of pentose sugar in case of DNA is deoxyribose sugar. In case of RNA, you have another ribose sugar which is called ribose sugar. Deoxy is not there, ribose sugar is there. Both are pentose sugar. Why pentose sugar? Because this sugar molecule consists of 5 carbon. If you're thinking about this particular corner, why this corner is not considered to be the 5th carbon? This corner have oxygen, not the carbon. That's why. Okay. So in this slide, you can see that bases are categorized into 2 types. You can see there are double ring structure. This is the single ring structure. So the double ring structures are called purines, and the single ring structures are called pyrimidines. These are the 2 types of nitrogenous base. You can check, you can pause this video and you can have a look for 1 minute to understand the difference between the double ring structure of nitrogenous base, which is called purines, and the single ring structure, pyrimidines. And all these bases, adenine, guanine, which comes under purine, cytosine, thymine, and uracil, which comes under pyrimidine, all are connected to ribose sugar or pentose sugar, you can say, or you can say deoxyribose sugar. But you have to remember, when I am saying deoxyribose sugar, I am talking about DNA. When I am saying ribose sugar, I am talking about the RNA molecule. So that you have to remember. Well, nucleic acid are polymers. So as I mentioned in the beginning, that nucleotide is a one unit. So there are so many nucleotides being attached, right? You can see here in this particular diagram. Like, let's see this one. A base, sugar and phosphate. This is one nucleotide. Another nucleotide is G, guanine attached to sugar and phosphate. These two nucleotides are again joined by phosphodiester bond. Always remember. Right? In the previous section, I have talked about this bond, this particular bond, which is phosphodiester bond. This bond is also phosphodiester bond, right? Which is connected to the third carbon of the pentose sugar. Now, in this particular sequence, you can find uracil is there. So when uracil is present, that means it is RNA molecule, right? Let me go back and tell you the difference. So in the thymine is only found in DNA. You will won't get in RNA molecule. In RNA you have uracil. You won't get DNA. You won't get thymine in the RNA molecule. But the common nitrogenous bases which are present in both is adenine, guanine and cytosine. So that will be there. But if uracil is mentioned, that means it is surely an RNA molecule. If thymine is mentioned, that means it is a DNA molecule. So in this slide, you can see uracil I have mentioned here. So that means it is a complete RNA molecule. And the sugar and the phosphate molecules make a rigid backbone for the structure of the molecule to retain. So the nucleic acids are, it's a polymer unit made up of a joining many nucleotide by the condensation reaction. When we say condensation reaction, the meaning is energy is going to release, right? And that energy, water molecule is going to release, and energy will be used up to make a bond, right? So the nucleotide is considered to be the monomer unit, and the nucleic acid will become the polymer. Like in this case, you can see in case of DNA, this is one nucleotide, this is another nucleotide chain, long chain, and both the chains are joined. That's why we call them double-stranded DNA. And IB can ask, they can give you this structure and they can ask you to circle one nucleotide. So you can see how I have circled it. You also circle in the same way in your exams. If they're asking you to, let's say, draw basic structure of DNA. So you can draw a small strand of DNA which contain AG and CT, AT and GC pairing, and you can mention everything like the bases, pentose sugar, phosphate, the bond, right? And the bond between the bases, which is a hydrogen bond actually. So between G and C, you can get triple hydrogen bond, and between A and T, you will get double hydrogen bond. This is called base pairing. Again, we will discuss about it in the next slide. So this is how the polymers are formed. By which reaction? Condensation reaction. What happens in the condensation reaction? Water is released. So whenever one bond is formed, one water molecule is released. Right? Okay. Now let's see the parts. Parts I have I've already discussed, but let me segregate it once again. So you will find three major parts. First is the nitrogenous base, right? Second is the pentose sugar. So as I mentioned, this particular end is not fifth carbon. This is the oxygen. So oxygen is shown here, and this is the phosphate molecule, which is interacting with other water molecules or any other polar compounds in the cell. So we have primarily five types of nitrogenous bases: adenine, thymine, cytosine, guanine, and uracil. And there is a five carbon sugar molecule which is called ribose in case of RNA, deoxyribose in case of DNA, and a phosphate group which is negatively charged. So the negatively charged on the phosphate group allow the DNA to interact with the histone proteins. So later on we are going to talk about how the DNA is wrapped around a nucleosome, which is a protein. So these charged molecules help the DNA to get wrapped around the protein, which is called nucleosome, and this coiling allows meters of the DNA to fit into a single cell. So imagine the length of the DNA and how they are going to get concise to a small area in the cell. These two are the covalent bond, right? This one is called glycosidic bond or covalent bond. This is phosphodiester bond or covalent bond. You can write any answers, both are correct. And these bases will they have a information what type of proteins are going to be formed. So it is a type in the sequence of the nitrogenous base in the DNA which forms a basis of genetic code. And how genetic code makes protein we'll discuss in some other topic. Not now. Okay, let's go ahead. So now the question what IB will ask. This is the basic structure of the nucleotide. IB would only ask you to draw a circle. Hexagon and rectangle to draw the block diagram of the nucleotide. So nobody will ask you to write the molecular structure. So phosphate group and nitrogenous base must be connected to the correct corner of the pentagon. This is very important where student will do mistake. So please take care of this. Let's talk about the pentose sugar. So pentose sugar means a sugar molecule which consists of 5 carbon, and pentose sugar is considered to be the monosaccharide, right? So if a monosaccharide have 5 carbon, we call them pentose. If they have a 6 carbon, we call them hexose, right? So this is how we can differentiate. So that we will study in the carbohydrate section. Now to identify ribose and deoxyribose, you can find the basic difference that in DNA pentose sugar, there is no oxygen on the second carbon, you can see. But in the second carbon of the ribose sugar, there is oxygen. Hydroxyl group is there, right? So nitrogenous base connect off to the carbon 1 in both, you can see. Second important point is carbon 5 is branched out to ring. They are connected to the phosphate group in both. And the phosphate group connects to the carbon 5. And if you look to the second carbon of the pentose sugar, you will find that there is hydroxyl group in the ribose sugar, but there is only hydrogen in case of deoxyribose sugar, right? So that gives a structural difference to the pentose sugar of DNA and RNA, or difference between ribose sugar and deoxyribose sugar. This is very important. So at the end, now we can say that RNA and DNA are a single strand. RNA is a single strand structure, DNA is double strand. Why double strand? Because both polynucleotide chain are held by base pairing, right? Both structure are helical. You can't say that RNA is not helical structure. It is helical structure, right? DNA is double stranded. The bases which are present in RNA is adenine, guanine, cytosine, and uracil. And in case of DNA, we have cytosine, guanine, adenine, and thymine. Both are joined by complementary base pairing. So I can say that adenine of strand polynucleotide strand 1 will bind with thymine of polynucleotide chain 2nd or another nucleotide chain. In this way, they are paired up, also called complementary base pairing. Now DNA structure, now you have understood. Let's be able to label these following in the structure. So I will wait for few seconds to see, and then we will do the practice. There is a 100% chance the question will come here in SL and in HL. It's compulsory. It's very important questions. So 5 prime and 3 prime end you should able to identify in a polynucleotide chain. Phosphate, which is generally circled. Deoxyribose sugar, which is also in the pentose side, right? Purine, pyrimidine, AG is purine, pyrimidine is CG. Sorry, not CG. AG is purine. CTU is pyrimidine. Deoxyribose sugar, you should able to number it. You can able to show the hydrogen bond, covalent bond, adenine, thymine, guanine, cytosine, and one nucleotide chain. So let's do practice. Let's try to label. So 5 prime end I have labeled. How? You can see my cursor. This is the first carbon, this is the second carbon, fourth carbon, and the fifth carbon is attached here. So of course this will be the 5 prime end. Let's see this one. First carbon, second carbon, third carbon have hydroxyl group. So this is free carbon. Why free carbon? Because nothing is attached here. So this is 3 prime end. So now you can able to identify 3 prime and 5 prime. Next, phosphate and deoxyribose sugar. This is deoxyribose sugar, pentose sugar, and this is the phosphate. I have circled it. Purines and pyrimidines. So purine, we know it's a single ring. Pyrimidine is a double ring. So you can choose any one and you can represent. If it is not given in the molecular form, but they will be written G, C, A, T, you can identify. Now deoxyribose sugar numbering, how to give the number. So this particular pentose sugar is numbered like this: 1, 2, 3, 4, 5. Hydrogen bond and the covalent bond. Hydrogen bond you will get in between the nitrogenous bases, always represented by a dotted line, and the covalent bond you will find between base and sugar and between sugar and phosphate. Always remember. How to identify adenine and thymine? Thymine is always a single ring. Now the question is how to identify, because adenine binds with thymine by double hydrogen bond. So this is how we can recognize. Guanine and cytosine, triple hydrogen bond. And the last important structure is shown in one square box, one nucleotide. Very, very important. So you can pause this video, and if you want to practice, you can practice. Meanwhile, we will go ahead. Let's talk about the DNA and RNA structure once again. So both DNA and RNA are the polymer of nucleotide, as we have discussed so far. However, you will find the difference between these two structures. What are the difference? Let's talk about the first difference. First difference, in DNA you have deoxyribose sugar, in RNA you have ribose sugar. Got it? This is the first difference. Now, let's talk about sugar phosphate bonding and the sugar phosphate backbone in case of DNA and in case of RNA molecule. How they are connected, how the bond is formed, how the structure is being stabilized, structure is being maintained. Let's talk about it. So nucleic acid, they have a backbone. When we say backbone means, see, structure is quite big. We need some support to the DNA molecule so that the strand can maintain their function. Because when we talk about function, structure is the main thing. If structure is not maintained, functionality will not going to come, or they will not be functional. So to make them functional, we have to maintain the structure. That is why the most important is the formation of backbone. So in DNA and RNA, there is a backbone, and that backbone is made up of phosphate, sugar, and then phosphate. You can see here. This is phosphate. Again, you have sugar molecules. Again, you have phosphate, this sugar molecule, again phosphate, sugar molecule. So there is a series of phosphate sugar, then again phosphate sugar, then again phosphate sugar, like that. And at the end, we have a 2 primes. The top one is 5' end because the 5th carbon is attached. Here the 3rd prime is free, so that's why 3 carbon. So the end of the backbone is always identified as 5' to 3'. What you have to remember here, 5' ends with phosphate and the 3' ends with pentose sugar. This is important because this concept will again come in the process of DNA replication, transcription. So we have to understand this backbone concept. So when the backbone is formed, actually, so what is happening? It is a reaction. It's a condensation reaction. You can see phosphate group is giving hydroxyl group, and the 3rd carbon have OH, which is giving hydrogen is coming out. So when hydrogen is coming out from here and hydroxide ion is coming from here, water molecule is formed, and you can see the phosphate is now directly connected to the oxygen of the sugar molecule. This is your phosphodiester bond, this one. And this condensation reaction is a type of a chemical reaction in which 2 molecules are joined to form a single molecule, and there is 1 water molecule lost. So this is an example of condensation reaction. So in the condensation reaction, what is happening? The 5' phosphate group of 1 nucleotide, let's take an example of this one, forms a new covalent bond with the 3rd prime carbon of the pentose sugar of the next nucleotide. So this is how they are connecting. So in this way, the backbone is formed from 5 to 3. You can check. This directionality has significance for DNA transcription and replication. So there is application of this. Why it is always 3 to 5 or 5 to 3. We'll talk about it in the another section of the topic once again. So backbone look like this. So backbones are like rungs of the ladder, and the nitrogenous base pairs act as a step. You can see it's like a ladder, right? So there are sugar phosphate backbone on one side, another side also have a sugar phosphate backbone, and in between you have a base pair. So base pair is acting as a steps for on the ladder, and both sugar phosphate backbones are running in opposite directions. So if you can see the orientation of the arrangement of the backbone, it is opposite. So that's why we call DNA is anti-parallel also, because of this nature, because of the opposite direction of the nucleotides, and that makes the structure twisted together to form a helical shape. So later on you can see the DNA is now coiled up to make a helical structure. What about RNA? Do they have a backbone? Of course they need a backbone, because the structure is not going to get stabilized. If the RNA strand is shorter length, It is possible that they can maintain their structure, but if it is quite long, there is a chance of base pairing also in case of RNA. Like in the messenger RNA, mRNA contains the information about the protein which is going to be formed in the cell. So the informations are mentioned in the form of a codon, and the tRNA is again a single strand RNA, but they have a binding sites also. You can see there are some bases of RNA molecule has been bind. This tRNA have anticodon, which is complementary to the codon of mRNA, and the function of tRNA is to bring the amino acid at a site of protein synthesis. This is our RNA molecule called ribosomal RNA, which is formed by the RNA molecule plus protein molecule. So you can see there are some RNA strand present in this particular ribosome. And all these non-genetic RNA is found by DNA itself. So DNA know what type of protein they have to form, where they have to do the protein synthesis, right? And who will bring the raw material. See the mastermind. I mean, everything is being already planned, right? So it makes the journey very easy for the DNA to express themselves, right? Like how we have a backbone in our body, the backbone maintains the structure of the human body. In the same way, to maintain the structure of the DNA, we need a backbone. That's why the sugar phosphate backbone is present in the DNA, which provide structural support and maintains the flexibility of the structure. So the sharing of electron in the covalent bond between the sugar and phosphate provide that strength to the structure due to the bond. This strength actually maintains the nucleotide in a specific orientation and in sequence which is necessary for storing, replicating and expressing the genetic information of the DNA. That's why backbone is very important. Bases in each nucleotide acid that forms base of the cone. Let's talk about the nitrogenous base. So we all know there is 5 types of nitrogenous base we have studied so far: adenine, thymine, cytosine, guanine and uracil. Whereas cytosine is single ring and cytosine molecules have amino group and CO group. Whereas uracil have oxy group, 2 oxy groups. Thymine have oxy group and 2 oxy group and methyl group. So the difference between uracil and thymine is just methyl group, which is present on this particular carbon, which is 1, 2, 3, 4, 5 carbon. 5th position. This is your. And in case of guanine, it's a double ring. First difference, it is a double ring. Second difference is amino group, oxy group, both are present. And in adenine, amino group is only present. So we know that A binds with T, right? So let's try to find how many bonds they are going to make, right? So there is a 2 oxy group in thymine. That means there is a possibility of making 2 hydrogen bond. In guanine and cytosine, there is a 3 possibility. There is oxy group, ammonia group, and hydrogen is in between the 2 nitrogen. That is also possible to make a hydrogen bond. So condition for the hydrogen bond is we need hydrogen. Second condition is the hydrogen must comes in between 2 electronegative elements. Like in this case, electronegative elements are oxygen and nitrogen. So whenever hydrogen will come, hydrogen bond will form between the bases, right? Okay, so this particular guanine, adenine found in both, they are double ring. Cytosine, thymine, uracil, they are the pyrimidines which are single ring structure, whereas uracil only found in RNA. Now let's talk about the sequence of the base which forms a code. I was talking about code, if you remember, in the mRNA molecule. So in order to in the order in which the different types of nucleotides are arranged in DNA or RNA, they serve as a code for the storing information for the living organism. But when we talk about serving as a code, code is a system in which one symbol signify a meaning of another symbol. Like the genetic code is a group of 3 nucleotide bases which is coded for a specific amino acid. Like, take an example. Let's see here. C, this is the first base. Second base is U. And third base is again U. It will code to leucine. C, U, C, again leucine. C, U, A, leucine. C, U, G, again leucine. C, C, U, proline. C, 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 proline. So this is how you can understand the language of the genetic code, right? Like you can see here, this is the DNA twisted form. One strand has been shown here. Once DNA strand is opened, you will find, like, make it straight and see the bases. So GCG, GGG, right? These all are the codons. 3 letters present on mRNA is called codons. So each codon is coded for a specific amino acid. And in this way, one amino acid combined with another amino acid to make a long chain of protein in the body by the process of translation. So this shows how the sequence of base forms a code and how the code is connected to the formation of protein in the body. And this is not end. This is the sequence of the bases forms code, which is a big data and very big and very specific data which is stored. So how this information is traveling? So DNA have the information. The DNA makes mRNA copy, means messenger RNA copy. So information will go from DNA to the mRNA. mRNA have the base sequences, which is called the three successive base sequences are called codon. For each codon, there is a specific amino acid coded for it. So the process of decoding a gene consists of 2 major steps, that is transcription, in which DNA is making an RNA copy, and then RNA copy will forms protein by translation. So this is the 2 step in which the information of the DNA is being decoded, right? See, it's not easy to take information from the DNA, right? So DNA needs a specific area in the cell where they will do the process. So transcription takes place in the nucleus and the translation takes place in the cytoplasm. So the gene, now what is gene? Gene is a specific sequence of the DNA that code for making a protein. And genes can vary from 100 to millions of nucleotide long. So it's not fixed. It's always change. So the diversity of the possible gene sequence and the length means there is a limitless capacity of DNA for storing genetic information. So this particular right side of the diagram shows the sequence of DNA nucleotide for insulin protein. Imagine, this is the sequence. If you have the sequence, you can produce insulin in the lab. Very easy. DNA has a double helix made of 2 anti-parallel strands of nucleotide with 2 strands linked by hydrogen bond between the complementary base pairing. So now you know that in DNA is a double helix, means there are 2 nucleotide chains running in opposite direction. That's why we call them anti-parallel. And both the strands are joined by hydrogen bond on the basis of complementary base pair. When we say complementary, means A will bind with T, G will bind with C, and vice versa. These are the bases. we know it. These are the backbone, we know it. And by the process of condensation, these polymers are joined, right? And they form a backbone. Helix are formed like this. When we talk about anti-parallel nature, the meaning is this, as we have discussed. So the nitrogenous bases will join together via hydrogen bond. You can see here. So between G and C, there is a triple hydrogen bond, and between A and T, there is double hydrogen bond. So the hydrogen bonds which is formed between the bases, that is always complementary. It's fixed. So this is your basic component of the nucleotide combined to make one nucleotide. Many nucleotides are joined to form a polynucleotide. Two polynucleotide chains are joined by hydrogen bond by the base pairing, and then later on it forms a structure. Let's talk about difference between DNA and RNA. You can expect question from here where they will ask you to write the difference. So first difference we both know, now we all know, ribonucleotide is a single strand, deoxyribonucleotide is a double strand. That's first difference. Second difference is the type of pentose sugar. So in case of DNA we have deoxyribose sugar, and in case of RNA we have ribose sugar, right? Third difference, base pairing is present in DNA between A, G and CT, whereas base pairing is not present in RNA molecule, right? So far we have discussed three differences. First, DNA is single strand, DNA is double strand, RNA is single strand. Second difference, DNA contain deoxyribose sugar, RNA contain ribose sugar. Third difference, the base pairing. Base pairing is present in DNA, absent in RNA. Next difference you can say, there is a two sugar phosphate backbone in DNA, whereas in RNA there is a one sugar phosphate backbone, right? So four differences now we have. Next, fifth difference, DNA contain thymine, whereas uracil is present in RNA. Next difference is the base pairing, that what we have discussed. So there is a possibility that there is a base pairing between the bases of the RNA, that is seen in the tRNA molecule, right? So you can mention except tRNA. In tRNA there is a chance of supercoiling, and there is a possibility of complementary base pairing. Another difference between the DNA and RNA is the function, right? So now we have 6 differences. DNA pass hereditary information between the generation of the cells, and they make RNA, and then it helps in protein synthesis. Whereas RNA, they make RNA by the process of transcription. Whereas in RNA molecule, they helps in translation, and they also help in making other types. Like other types of RNA molecule are mRNA, tRNA, rRNA, which helps in protein synthesis. Next very fundamental difference is, most DNA is confined to the nucleus, but there are the DNA molecule which is present in cell organelle also, like mitochondrial DNA, chloroplastidyl DNA, which is present in the chloroplast. Mitochondrial DNA is present in mitochondria. But there are RNA molecule which is only present in nucleus. This is also an example you can add as a difference. DNA is also found in prokaryotes, right, in the nucleoid region, whereas RNA is freely found in the cytoplasm of the bacteria. So 6 or 7 differences we have seen so far. Let me recap once again. Starting with the first difference. First difference, DNA is double-stranded, RNA is single-stranded. Second difference, DNA contain deoxyribose sugar, RNA contain ribose sugar. Third difference, there is a base pairing in DNA, no base pairing in RNA. 2 sugar phosphate backbone are present in DNA, 1 sugar phosphate backbone are present in DNA. Fifth, thymine is present in DNA, uracil is present in RNA. Sixth, complementary base pairing you can discuss. Seventh, function. DNA helps in passing the information and making RNA. RNA function is to make protein. Seventh difference is DNA is present in cell organelle as well as nucleus, whereas RNA is only found in the nucleus, which is formed by transcription. So there is a possibility of getting 5 or 6 marks question from here. Write the difference. Right. Now let's talk about the role of complementary base pairing in allowing the genetic information to be replicated and expressed. What is the purpose? What is the importance of this complementary base pairing? Let's talk about that. So these are the bases. So the nitrogenous base joined together via hydrogen bond we have seen in the structure. So when we talk about the base pairing up, you will find there is a consistent order which is being followed in the base pairing. A binds with T with double hydrogen bond. G binds, then in case of RNA, there is a possibility A binds with U with 2 hydrogen bond. Guanine binds with cytosine with 3 hydrogen bond. This is possible complementary base pairing. So in DNA, the nitrogenous base for hydrogen bond with complementary bases paired as A to T and G to C. A to T by double hydrogen bond, G to C triple hydrogen bond. In case of RNA, uracil replaces thymine. So the complementary base pairing becomes A to U and C to G. Right? A to U will be double hydrogen bond, G to C is triple hydrogen bond. And the complementary base pairing have an important role in the allowing genetic information to be replicated and expressed later on. So that expression things we will study in D topic. Even through RNA is a single strand, there is a possibility that there is a complementary base pairing, because if the RNA strand is quite big, right, you can't maintain that thread, a long thread. So in the 3D shape, there is a possibility they can merge, they can come closer and they can form a bond. So that is also possible. Complementary base pairing during the process of DNA replication. Let's talk about that. This particular DNA, which was double strand, it's now opened, right? So you can see the base sequence, and the new bases has been added in both the strands, and now we have a 2 DNA in the file. Now let's try to match the DNA constituent, the base sequence from the parent DNA. AT, GC, GC, TA. AT, GC, CG and TA. So it's same. So the DNA replication occurs before the cell division, and it is required for reproduction and the growth of the cells and the tissue, so that they can maintain growth and they can replace all the dead cells. So DNA replication becomes very fundamental process to maintain the consistency in the information, meaning that original strand will be there in the new strand. That is why the DNA replication is semi-conservative. When we say semi-conservative, 50% of the DNA will be new, 50% will be the newly formed. Old one, 50% is newly formed. So the enzyme, DNA polymerase, they helps in building all these structures in the cell, and this results in the formation of the new strand having the same sequence of DNA, same sequence of bases. In this way, DNA replication builds 2 identical copy of DNA, thanks to DNA polymerase 3 who is working this. Again, the base pairing is happening in the transcription process. So during transcription, DNA strand will open, and one of the DNA strand will act as a template surface for the formation of the mRNA molecule. So the sequence of DNA nucleotide is the base for genetic code used for production of proteins and RNA molecule. So the instructions stored in the gene of the DNA are expressed in two steps. First, transcription will take place, in which mRNA copy will form, and then it is undergo translation when protein is going to form. So transcription is the synthesis of RNA using DNA template. So in the diagram you can see this is the DNA template. Here we need an enzyme called RNA polymerase, which is going to build an RNA molecule on the template strand. And what are the bases? You can see exactly complementary. And the transcription will result in the RNA strand having the complementary sequence of the base as DNA. So it's maintained the information stored in the sequence of nucleotide of the code. So information is not changed. So that's the importance of the complementary base pairing. When we will make a protein, that's again a good example. This is mRNA, which is here, this one. So you can see my cursor. This is tRNA. If you remember, I told mRNA have codons and tRNA have anticodons. Both are complementary. And see, each tRNA molecule is bringing one amino acid in the process. So translation is a formation of a polypeptide chain from mRNA molecule. Where this process takes place? On the ribosome. So ribosome builds the polypeptide chain by reading the information on mRNA, binding the coded amino acid to the polypeptide chain simultaneously. And in this way, a long chain of proteins are formed by the temporary complementary base pairing between codon and anticodon, and in this way a long chain of proteins are formed. Looks very sophisticated step, but yes, it is very sophisticated and very precise function done by the DNA throughout the process to produce a functional protein in your body. Diversity of the possible DNA base sequence and the limitless capacity of the DNA for storing information. Let's talk about that. So when we say DNA stores, what they are storing? Storing for future use. Information, what information? The knowledge which is going to show the effect in the body. When we say about sequence, means the exact copy we need. Copy of what? Copy of the nitrogenous bases. Because these things are going to work in the body to produce protein in the cell. So information is passing from generation to generation and proteins are produced continuously. So this cycle is keep on going. And when we talk about the limitless, we are talking about each nucleotide of the DNA have the 4 nitrogenous bases. So these bases can be in any order. So you can't justify which order is fixed. So DNA strand of any length. Therefore, in a DNA strand, n base length are there. So possible 4 to the power n possible sequences. So let's say the DNA shows small section of DNA with 15 nucleotide base pair. In this example, there will be 4 to the power 15 possibilities of DNA sequences. That means this much possibility. Which one you have? We don't know. Imagine. So DNA can store a staggering amount of information in an almost inconceivably and in very limited volume, very small volume they can store. While still in early stage of this computational development of science, computer science scientists are developing a way to use DNA information in a digital information. They can feed all this information in the computers, smart devices, television, thermostats, home security systems, digital personal assistant, wearable cars, robots, and other devices that are generating and using data in exponential increasing amount. So now DNA is being very versatile role because of the storage capacity, because of the uniqueness, right? And the storage capacity. So the information are converted in so many, like the DNA sequence is taken bases, then decoded into the binary codes 0 and 1, and then by the binary code it is converted to readings in a digital file, and that can be used. Conservation of the genetic code across all life forms as evidence of universal common ancestry. So I've discussed about this in the beginning in very basic. Let's talk about it once again. So how we can say that it's unique and we all are coming from common ancestor? In order in which the different types of nucleotide are arranged in a DNA or in RNA, this serves as a code for making a protein. And the process of decoding these gene consists of two major steps: transcription and then translation. This is how they can maintain the life. And when we talk about the universality of the genetic code, means it is universal. Like, let's take an example. If I'm saying AUG is coded for methionine, if AUG code is present in bacteria or in fungi or in plant, animal, insect, everywhere, it is coded to methionine. So genetic code is very highly universal. Everywhere you will find the same. So the genetic code is universal, which means that with just a few variation, all living organisms use this same genetic code. Even virus use this same genetic code to produce anything. So the different organism might have a different sequence of nucleotide, but the decoding of the sequence is done using the same code. So we don't have a different dictionary for that. So this is the language of the DNA, the dictionary which we are talking about. For example, let's take an example of GUA. This is G, this is U, and this is A. Valid for valine. So valine in animal, in plants, or in bacteria or cyanobacteria, everyone have the same valine only. So it's not going to change. That's what they defines universality of the genetic code. It's universal. Evidence of common ancestry. Biologically and chemically, there is no reason why this particular genetic code should exist, rather than any of the theoretical possible others. Yet with one minor variation, each species on Earth uses the same genetic code. The explanation of this universality is that the gene code was the one carried out by the last universal common ancestor of life. That's Luca. And it has been passed on over time to all of its descendants. And that is how still we are following the same genetic possibilities. Now we have completed the SL and HL common topic. Now we are strictly going to start HL section. You can see here it's mentioned HL. The first topic is directionality of the RNA and the DNA. So here we will talk about in slight more detail about the sequence, about the numbering of the carbon. So nucleotide are consist of purines and pyrimidines, which is called nitrogenous base, pentose sugar, and the phosphate. Right? Pentose sugar have 5 carbon. Right? 4 of the carbon plus an oxygen makes a ring. You can see 1, 2, 3, 4 the carbon and 1 oxygen in a ring, and the shape looks like pentagon. So to identify the carbon, there must be numbers sequentially like this: 1, 2, 3, 4, and 5. Right? Moving clockwise from oxygen onward, like from the 0, from oxygen within the ring. So leave the oxygen, first carbon, second carbon, third, fourth, like that you can count. So 5th carbon connects with the phosphate group by phosphodiester bond. Of course, they have a backbone. We have discussed about this in the backbone section. DNA is antiparallel. This also we have discussed. Directionality, like in the 5 to 3 directionality of the DNA, RNA affects the way enzyme can build to and function when bound to the nucleic acid. So DNA polymerase enzyme works during DNA replication process, whereas RNA polymerase works during transcription process and ribosomes during translation. So their structure, their directions play a major role in fitting with the enzyme, right? So if enzyme is not fitting, that means they can't work on it. So in case of DNA replication, a parent strand is going to make one more copy of the DNA strand, and enzymes that build the DNA during replication is DNA polymerase, which adds nucleotide in 3' end. If you have a question why 3', why here, you can see my cursor. Because 3' have hydroxyl group, which is a functional group. So when you want to add another nucleotide, you can't add here at 5'. You can only add it to the OH group. Why? Because the 3' OH group will give hydrogen, and the phosphate have OH group, they will combine and the water molecule is released and phosphodiester bond is formed. So the prime at which length will grow, the base length will grow is 3'. You can add new nucleotide only at 3' end of the DNA strand, not on the 5' end. So in this way you will get that a leading strand is formed and as well as the lagging strand is formed. So since both the DNA strand is being used in the process of DNA replication, so there are differences in how the process is occurring on both the strand. So on one strand the direction is fixed, 5 to 3. So DNA replication going in the leading form, like it will keep on forming as the DNA is getting open, the process is continuously going. But there is another strand in which there is a discontinuous manner called lagging strand. So the new DNA polymer 3' end is opposite to the opposite direction of the replication. That is why it is opposite. But in case of transcription, DNA have to open, and in this case only one strand will act as a template strand. In the previous case both were acting, but now only one strand will act as a template surface. So the enzyme for transcription is RNA polymerase, can only add nucleotide to the 3' end of the growing polymer of RNA nucleotide. So this is 3'. So you can add here only because the 5' is already out. You can't add anything else because 5' is already being occupied by phosphate. Right? So you can't add anything else there. You can only add here at the 3'. So the 5' phosphate ends is added to the 3' ribose end of the growing RNA strand. And in this way, the direction of the transcription is 5 to 3'. You can see the diagram. But in case of translation, what is happening? Translation is a synthesis of a protein from the mRNA. So during initiation of the process, the ribosome attached to the 5' end of the mRNA. So this is 5'. So ribosome is attached to this particular prime. And ribosome then move along the mRNA towards the 3' end. And in this way, translation direction is 5 to 3. Like this. So direction is moving this side. So you can say that ribosome is moving towards the 3' end of the mRNA so that the process can go on. This is the direction. Purine to pyrimidine bonding as a component of DNA helix stability. Let's talk about how they are maintaining stability. So these are the bases we have. These are the structural bases. These are found in the DNA and the RNA. But when it comes to the combining both the bases, you will find that G binds with C. And as I mentioned, for hydrogen bond formation, you need hydrogen first, and second is electronegative element. So when hydrogen comes in between two electronegative element, hydrogen bond is going to form. So here, this hydrogen comes between this oxygen and this nitrogen. Both are electronegative, hydrogen bond is formed. Here also, this hydrogen is coming between two nitrogen. This hydrogen coming between nitrogen and oxygen. So there is a possibility of 3 hydrogen bond. But in case of adenine and thymine, Hydrogen comes between nitrogen and oxygen. Here hydrogen comes between nitrogen and nitrogen. The third one is wrong dotted, so you can please ignore it. This is not possible, right? Like this, you can see this is the another diagram. This is the bond length, you can see the diameter of that particular base pair is nearby same, you can see 1.08 nm. So they are maintaining the consistency in the width-wise consistency. Now the question is how this big data of the DNA is being incorporated in very small space. So nucleosome, like how we in a storehouse, the things are kept in a box, right? So storage unit is an analogous to the nucleus. So boxes are analogous to the individual chromosome of the DNA, and the things stored in the boxes are analogous to the DNA nucleotide. So imagine this situation. So in eukaryotes, DNA is packed and stored in the nucleus. So the quantity of DNA to pack, the diploid human genome contains approximately 6 billion base pairs. Imagine. And all these 6 billion base pair is packed into 23 pairs of chromosome. So total 56. Sorry, 46, not 56, 46. Because each base pair is around 0.3 nanometer long, right? Each diploid cell therefore contains minimum 2 meters of DNA. Moreover, it is estimated that the human body contains about 50 trillion cells, which works out to about 100 trillion meters of DNA per human. Wow. And 100 trillion meters of DNA means each of us have enough DNA to go from Earth to Sun and back more than 300 times, or around the Earth equator 2.5 million times. Imagine, how big DNA length and the data we have. When we talk about packaging, it's even if you have ever tried to store a garden hose, you know that it is much easier to do so if you bring by coiling the hose, right? Hose. So the same thing happened with the eukaryotic DNA. It has layer of coils that compact the molecule so that it can fit into the small area like this. So this the DNA is being wrapped around these beads, right? And these beads are making an octamer, 8 units. So if you can count, it will be 8 unit. So between two nucleosomes, there is a linker DNA, right? Nucleosome have 10 nanometer diameter, which thing is how they are packed, and they are making a 700 nanometer of the supercoiling. Wow. And how the pack look like? This is one nucleosome in which there are 8 proteins. H2A, 2 molecule, they are in the dimer form. So 4 protein molecules which are present in dimer form, it becomes octamer. So the basic structural unit of DNA packing is nucleosome. This is nucleosome, the diagram which shows. And the nucleosome consists of a length of a DNA coiled around the core of 8 histone proteins, right? You can count 1, 2, 3, 4, 5, 6, 7, 8. And this protein is called histone protein. Histone are the family of the small positively charged protein. And we know that phosphate have a negative charge. So that charge is being cancelled by the positively charged protein. So in this way, DNA becomes neutral. So due to phosphate group in the nucleotide, DNA is negatively charged. DNA binds tightly to the histone protein and make use of electromagnetic attraction and helps to neutralize the charge. And there's H1 protein attached to the adjacent nucleosomes and bind to the DNA nucleosome's core so that they can bring it near. And in this way, one octamer connected to another octamer like this in a long chain, like this. This is the molecular diagram. DNA packing. So in this way, the DNA is packed very compactly arranged, and nucleosome is playing a major role here. And in between the nucleosome, there are the linker DNA. So each chromosome have hundreds of thousands of nucleosomes, and each chromosome is thus a long chain of nucleosomes, which gives an appearance of a strings of beads when views using an electron microscope. And this is how they are arranged. So DNA packing starts during prophase of mitosis or meiosis. The DNA condenses, right? So supercoiling takes place, and condensing of the DNA into chromosome prevents the tangling and the damaging of the DNA content during cell division. So it's become very important to maintain the structure. But when we talk about the DNA unpacking, so unpacking is done in a very sequential order. First, disruption. In case of disruption, histone protein will get disassembled, then recycling, right? And then a new histone protein is formed. So process such as transcription and replication requires the DNA to temporarily release from the histone. DNA will return to its compact state after that. So it's not like DNA is open to do the let's say do the replication. After that, they are not going to get coiled. Of course, they will get coiled and they will go back into the original state. DNA packing is again unique in both. Evidences how we have proven that DNA acting as a genetic material. Let's talk about that. So there are multiple concepts, multiple ideas given by many scientists during that time, and they have done lots of experiment. There was an experiment called transformation experiment, which was done by Frederick Griffith, scientist. Then later on, Robert Brown was a Scottish botanist whose pioneering the use of microscope lead to one of the earliest detailed description of the cell nucleus. He was responsible for that. Then German botanist Schleiden, who has proposed that nucleus play a major role in generating the cells. Then nucleic acid was isolated from the DNA by the first Friedrich, he is the scientist. Then Oscar Hertwig, he has discussed about the nucleus has an important role in the hereditary. Then chromosomal theory of inheritance was given by Walter and Boveri. There's a fruit fly Drosophila experiment was done by T.H. Morgan, Thomas Hunt Morgan, and he's also called father of the modern genetics. Is the genetic material DNA or protein? So there was lots of debate, lots of experiment done during that time to give the real idea. So the best one is Huxley and Chase experiment, in which they have seen the component of the cell. They found that the major confusion was between amino acid, between the protein and the DNA, which is acting as a genetic material during early 20th century. So this is American scientist Oswald Avery, has a microbiologist who research helped ascertain that DNA is the substance responsible for heredity, and studied called transformation of the bacterial cells. He has done a transformation experiment where he has taken two variety of that particular bacteria which cause disease, and then it was injected to the mice and they checked. Later on, using bacteriophage virus, Alfred Huxley and Martha Chase designed an experiment to solidify that DNA is the genetic material, and they've done very smart work, smart job. So in the virus, we know there is two things, DNA and protein. So first they have labeled protein, they have done the experiment, and the same experiment was repeated for DNA also. What is bacteriophage? Bacteriophages, when virus infect bacteria, and the structure called bacteriophages. So in the genetic material, the confusion was between RNA and the protein. So what both scientists have done, they have taken radioactive isotope and they have tagged the atom. Now when we say isotope, isotopes are generally radioactive substance, which means the nucleus is unstable and they are continuously releasing radiations, and this energy is being detected as a radiation, right? So what they have done, DNA contains phosphorus, so they have used radioactive phosphorus to tag that particular DNA, and sulfur is found in protein, so they have tagged protein also, right, in the bacteriophage. Means the DNA of the bacteriophage was labeled with phosphorus 32, which is a radioactive, and another sets of bacteria, they have labeled sulfur of the protein, which S35, which is a radioactive. And the sequence of experiments start. So first they have taken radioactive labeled protein in the cell. Cell was allowed to grow, then it was centrifuged. At the end they have found that there is no radioactive protein in the next generation, which proves that yes, they are not acting as a genetic material. That's why it's not gone to the next generation. If it is going to the next generation, that means it is acting as a genetic material, right? Because we know that in the new DNA form, one is old strand. So if one is coming from the old side, that means old side is already radioactive. So it must come, but it is not coming here. In the second set of experiment, this time DNA was tagged. DNA was tagged with radioactive phosphate. Same thing, infected on the bacteria. Bacteria have used the DNA. They have grown their population. Then it was taken out, blended. And this time, they have found that all the bacteria, the new bacteria, they have phosphorus 32. That means it's a clear evidence that it is radioactive and they have been passed on. That means DNA acting as a genetic material. So these are a series of reactions happen. They have done so many times to get a clear idea. So first they have attached that particular bacteriophage to the bacteria. Then step 2, the genetic material are released, which is called penetration. Then there is this biosynthesis of the genetic information. Assembly is taking place and then they are releasing it. And then scientists are checking whether they have radioactive phosphorus or radioactive sulfur. So radioactive sulfur was not possible, but yes, they have got radioactive phosphorus in the DNA. Chargaff data on the relative amount of pyrimidine and purine bases across diverse life forms. So it is now understood that DNA is a double helix made up of 2 anti-parallel strands, and both strands are linked by hydrogen bond. And the discovery of the structure of the DNA was not the work of a single scientist, rather than it is a collaborative result by multiple research working over multiple places for multiple decades. And there are lots of people involved in this. So nucleic acid was isolated from the nucleus in a variety of eukaryotic cell by Friedrich Mischer. Even though he had discovered DNA, it was not for nearly around 80 years that the structure and the role of DNA as a genetic material was determined. So imagine 80 years back, it was already, somebody has talked about it. Isolation of nitrogenous base was done by Kossel, the German biochemist. They have investigated the chemical composition of the nucleic acid between 8085 and 19, and they isolated the 5 types of nitrogenous base for the first time. Nucleotide was discovered by American biochemist. Tetranucleotide hypothesis. So Phoebus was the first to state that DNA molecule consists of nucleotide connected by 5 to 3 prime sugar phosphate bond with base attached to first carbon of the sugar. That they have given a hypothesis. But at that time, there was misarrangement of the molecule. So although the correct about the bonding of the nucleotide, the idea about the structure was wrong. The bonding pattern was correct, but the arrangement, the structure was wrong. So in 1909, he proposed tetranucleotide hypothesis, in which DNA was composed of 4 nucleotides per molecule, with 4 bases present in an equal number and directed to the outside of the structure like this, which is not possible, right? Then later on, there's a Florence, British scientist, who have done X-ray crystallography to study the DNA. In 1938, she and Williams Astbury published a paper that described the structure of DNA as a piles of pennies with a close succession of flat or flattish nucleotides standing out like this. But it was not convincing many people at that time. Frequency of nitrogenous base was done by Chargaff. So in the early proposal of the DNA structure, there was the implication that the four bases found in DNA were present in equal amount. However, in 1948, Erwin Chargaff shows that four sequences were not equal within or between species. It's keep on changing. So you can see AT sequence and GC sequence are different. It's not make sense. Franklin, she has done a very remarkable job in 1952, just 4 years before Watson, when he has discovered the DNA structure. She was the first who have given her photograph, the crystallography image of the DNA, and which shows that DNA is a double helix with the strand running in the anti-parallel direction. And her photo was photo 51. Triple strand model of the DNA, again given by Pauling, American biochemist who has already determined how amino acid bond to form a polypeptide, proposed a model for the structure of DNA. He had not seen Franklin evidence that DNA was a double helix at that time. Chargaff data suggests base pairing. Pauling models proposed that DNA was triple helix with the phosphate backbone on the on the inside and the nucleotides pointing outside, which was not correct, supporting the evidence. So the Pauling model of the DNA was quickly disproven, as the negatively charged phosphate will repel other things. So how they will maintain the structure? So that was a big controversy at that time. Later on, it was not convinced. Double helix model the DNA given by James Watson and Francis Crick use the model building, and they have generated the model of the DNA, and they have published their report in Nature in April 25th, 1953. So these are the evidences from his research paper. Now we know the base pairing. Now we know the each component of the DNA. Thank you for watching my video, and soon I am going to upload topic wise questions bank also. And if you want to need more question bank, you can contact me. And thank you for being patient and listening to this wonderful notes. This will help you for your exam preparation. Thank you, everyone.